Hey y'all, my name is Prashant Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're going to be doing a passage walkthrough in the Biochem section. You should be taking roughly 8 minutes on this passage, but here we're going to be going slow, taking our time, so we can see exactly what's going on in my head as we take on this passage. Let's get it. All right, great. So I've gone ahead and put the passage on the left and all 5 questions we're going through on the right. So feel free to take a moment and pause the video, go at your own pace, answer the questions, and then come back to us. If not, let's get right into strategy. So for me, I like to have the same strategy as I come into every passage. This gives me something to come to, even if the passage is easy or if it's hard, right? We want to have that same sort of mentality going into any passage, so we're not overestimating it or underestimating it. For me, that's writing a 3 to 5 word summary after reading each paragraph, going paragraph by paragraph. And the reason I like to do this is it provides me accountability, right? Oftentimes I would be reading through these paragraphs, not understanding what's going on, and feeling so lost at the end of the passage that I had to reread all over again, right? I call this reading words emptily. Instead, by doing this, we have like a quick 3 to 5 word check-in to make sure we understand the paragraph. That way, if we don't understand it, we can just reread the paragraph as opposed to rereading the entire passage, right? So writing this quick 3 to 5 word summary in your own words really helps hammer that in. The second tip I recommend is not spending too much time on the figures on that first read. The reason I really like this strategy is we don't know what the questions are going to ask about the figures, right? And it doesn't make sense to waste any time looking at a figure, trying to figure it out, especially if a question might not even ask about it. Instead, let's take a look at it, understand the big picture maybe, and come back to it if a question asks, right? We're probably going to have to come back to it anyways. Might as well only do that if the question asks for it. All right, let's get right into it. Extracellular phosphate, PI, regulates its own renal excretion by eliciting concentration-dependent secretion of parathyroid hormone. The mechanism by which phosphate is sensed, however, remains unknown. The calcium sensor receptor, CASR, is the main controller of PTH secretion, and researchers hypothesized that increased phosphate might have an effect on CASR. So what is happening, right? That's the question I want you to ask at each paragraph. And write a 3 to 5 word summary, right? And so I'm going to write PTH effect on PI excretion, right? This is the main point taken, and I want to put a little symbol or whatever you want to call it, right? And we don't know what CASR does for sure, but that's somewhere involved in this sort of mechanism. It doesn't really have to make sense to anyone else, right? No one else is going to be reading these. No one else is going to be looking at our notes. So as long as it makes sense to you, that's all we need. 3 to 5 words, some symbols, anything that makes it quick and understandable for you. All right, let's go on to the next paragraph. In experiment 1, researchers tested the effect of extracellular phosphate on the ability of CASR to induce PERK, one of its downstream targets. R568 was added to reduce the risk of calcium phosphate association in the presence of increased phosphate concentrations. In the study, researchers increased extracellular phosphate levels from 0 millimeters to 2 millimolars. The results are shown in figure 1, right? And so what's the question? What's happening? And here we have an experiment, right? We're looking at this phosphate effect on CASR and that's ability to induce PERK, right? I'm not going to go into every detail. I'm not thinking about, what does PERK do? What does CASR do? What does R568 do? All of that, right? We don't really care. All we need to know is there's an experimental design. If you want, you can circle this and call this the independent variable, whatever. We're focused on the big picture idea, and here we have an experiment. Next, we come across a figure. And what did I say? We're not going to spend too much time looking at the figure because we don't know what's important. All we see is effect of extracellular phosphate on PERK expression levels by Western blotting, right? A Western blot, simply put. And so we'll come back to that if a question asks for it, but we're really not going to stress too much about it. Western blots are complicated as is, and they don't look too great. Let's not waste any time looking at things we don't need to. Next, in experiment 2, researchers measured enzymatic kinetics and demonstrated that PI inhibits Caser in a non-competitive manner. The max response percentage of Caser was measured at increasing Ca2+ concentrations in the presence or absence of extracellular pi. The results are shown in figure 2. What's happening? Right away, I see one little phrase that stands out as pretty important, right? This PI inhibits Caser. And I'm going to make sure I write non-competitive because I know the MCAT likes to test enzyme kinetics and competitive inhibition and all of that, right? So just something to keep me focused on it being non-competitive inhibition. And I write this little flat arrow to show that it's inhibiting. Great. And then we come across another curve, right? And we're not going to stress too much about it, right? We see sort of enzyme kinetics, all of these curves that we've seen before. Let's not stress about it right now. All right. In experiment 3, researchers investigated the effect of PI on PTH secretion. Increasing levels of PI were added at a constant calcium concentration, and PTH secretion was measured. The results from the study are shown in figure 3. Okay, we have another experiment, right? And we're looking at increased PI effect on PTH secretion, right? Very simple. Not going to focus on the details. And look, we have another graph, right? And we're not going to stress too much about it. What do we see? We see sort of this independent variable of PI, dependent variable of PTH, this sort of box plot that's increasing. Not going to stress about it right now. Let's go on to the final paragraph. Researchers also showed that a mutation of residue R62 and anion binding site 1 abolished phosphate-induced inhibition of Caser, right? I'm only going to write something pretty brief, right? R62, important, right? For PI inhibition or something like that. It doesn't have to be too focused on the details. I had told you, I don't like focusing on the details on that first read, but here it's a small paragraph focused on this one R62 residue, so might as well pay attention a little bit. All right, so right away, we've gotten a sense for this entire passage. We're not stressed. We understand each paragraph in a broad perspective, not going into the details too much, but we have a great understanding to go straight into questions. So that's what we'll do. So I've gone ahead and put question 1 here. A patient with overactive PTH presents to a clinic. The doctor is most likely to prescribe which of the following hormones? A, epinephrine, B, calcitonin, C, thyroid stimulating hormone, D, cortisol. All right, so right away, this is a standalone question, right? It gives you the situation of a patient with overactive PTH, and we're looking to give him some sort of hormone. And so we don't need the passage for this, so let's go through the answer choices, right? Well, we know PTH has to do with something very specific, right? It has to do with the calcium levels. And what does parathyroid hormone, PTH, have to do with calcium levels? It increases calcium levels, right? And so we should know this is in a pair with another hormone, right? I'm going to write this down. PTH increases calcium, 2+. What hormone in the body decreases calcium? And this is something we should know from our content review phase, and that is calcitonin. Calcitonin decreases Ca2+. And a nice little mnemonic or way to remember this is that calcitonin tones down the calcium levels, right? A little silly thing to help you remember it, but gives us the answer right away, and we're at B. Let's go through the other hormones just to make sure. Epinephrine, dealing with stress, not really dealing with calcium levels. Same thing with cortisol. So we can eliminate A and D immediately. Thyroid stimulating hormone has to do with metabolism and not really as much dealing with the calcium levels. So we can eliminate that. And right away, we could have gone to B, but just so you guys know, we can always use this process of elimination to help get us to that answer. Great. So we're 1 for 1. Great work. Let's go on to question number 2. The researchers include beta-actin levels in figure 1 in order to A, control for cell type, B, control for pH levels, C, control for the amount of protein loaded in the gel, and D, control for unwanted proteins. Okay, so right away we have this figure where we're pointed to, figure 1, and that's this guy right here, this Western blot. And so the question is asking about beta-actin levels. And this is also a combination of sort of a standalone understanding as well as looking at this table, right? This Western blot is specifically focused on PERK expression, right? And PERK is this thing we're looking at in terms of Cassar and phosphate levels, right? But why is beta-actin involved? And in this case, beta-actin is a commonly used control. What type of control? It's called a loading control. Why do we use this in blotting? Well, we use this because expression of beta-actin is not changed very often during any sort of chemical reaction, right? And so why would we use it if it's not changed? Well, it helps indicate how much sort of protein we're putting into our Western blotting system. If we're putting too much, we'll see a heavier blot of beta-actin. If we're putting too little, we'll see a light blot. And so having this gives us like a check, right? We're checking if we have the right amount of volume of this sample of this protein in the gel. That way, if we see a heavier sort of blot in our the PERK expression, right? We're positive that it's not because we put extra sample in. We're positive it's because there's sort of increased expression. So think of beta-actin as a way to check for the correct quantity of something being added, right? And so which answer choice indicates that, right? It's not cell type, it's not pH levels, it's C, controlling for the amount of protein loaded in the gel. And it's not controlling for unwanted protein. So this is an important sort of control part of an experiment that's very helpful in Western blotting. And I would definitely recommend noting beta-actin levels. So we're 2 for 2. That one's a tough sort of standalone question that also combines some sort of knowledge seen in the passage. Let's go on to question 3. Lower pH leads to protein denaturation. Which organelle employs a lower pH to denature proteins? Is it A, protease? Is it B, mitochondria? Is it C, lysosome? Or D, nucleus? So again, we have a sort of standalone question, right? It deals with denaturing proteins, and we should be thinking something in our head right now, and it's in answer choice C, right? Lysosome. Lysosome are very, very common organelles that are used by by destroying proteins, and they're kind of like the killer of the cell, right? Mitochondria, that powerhouse of the cell, doesn't really make sense. Employing a lower pH to denature proteins. Nucleus doesn't really make sense either. Protease, while it's sometimes used to denature proteins, it is not an organelle, so we can eliminate A too pretty easily, and we're stuck with C. Nothing too challenging right here, and we're 3 for 3. All right, question number 4. According to the passage, when researchers add PI to cells, what is the most likely effect on the Km and Vmax of Cassar? A, they both stay the same. B, Km stays the same, Vmax decreases. C, Km increases, Vmax decreases. D, Km stays the same, Vmax increases. All right, so this is a very, very common type of question, right? Whenever we see Km or Vmax on the MCAT, we should be thinking of inhibitors or Michaelis-Menten, all these types of things, right? And so we noticed something in our sort of outline. We don't have to go back there because we probably remember it, but it's the fact that PI inhibits Cassar non-competitively, right? This is very, very important, so I'll highlight it. Non-competitive manner. And what do we know about non-competitive inhibitors, right? Well, we have the sort of enzyme binding to its substrate. I'll put this guy in S and this guy E, and it's binding, right? And the non-competitive inhibitor binds to the enzyme itself alone, but it also binds to the enzyme substrate complex, right? And they bind equally. This is why it's so special. It binds to the enzyme alone and the enzyme substrate complex equally. And so for this reason, Km does what? It stays the same. So we can eliminate A and C immediately. Why does it stay the same, right? Km shows affinity. And if affinity is not changing because it's binding equally to the enzyme substrate as well as the enzyme alone, then it won't change. Now we have to ask, what happens to Vmax? Does it increase or decrease, right? And so Vmax is this indicator of maximum sort of reaction happening. If the enzyme substrate is bound in any case, right, we know that Vmax is going to decrease. And so B is the best answer. It is very simple if you think about, as long as the substrate and enzyme are binding, but this inhibitor just adds to it, that complex goes away and it's not useful. So our Vm goes out the window. It goes down because it becomes useless, right? Even if the enzyme and substrate bind, it becomes useless. So right away, we are at B and we're at 4 for 4 and we're ready to go on to the last question. Okay. Oops. Based on the information from the passage, what is the most likely reason that the R62 mutation of Caser abolishes phosphate-induced inhibition of Caser? Is it A, R62 is crucial to protein folding, B, R62 interacts with phosphate in the anion binding pocket, C, R62 facilitates glutamate interaction with phosphate in the anion binding pocket, or D, is R62 creates the only anion binding pocket in the protein? Okay, so this is a challenging question where we're going to have to go back into the passage to look. But we know exactly where to look, right? And that's this final passage paragraph right here, because that's where it talks about R62. It says, researchers also showed that a mutation of residue R62 and anion binding site 1 abolished phosphate-induced inhibition of Cas-R, exactly what the question is asking for. So we know where to go. We didn't even need our outline, but our outline helps us hone in right there. Okay, so the first question we should be asking is, what does R62 mean, right? It's our residue changing. And we should always know whenever we have like this sort of letter followed by sort of number like this, we have some sort of change of a residue. And it tells you right here, we have a mutation of a residue. This first letter gives us the abbreviation of amino acid, right? And this number gives us the location. And oftentimes, if they give us a letter after it, it's what it's changing to. So amino acid changed to. Okay, so we have this three-part structure, but here it only tells us that R62 is changing. So R is changing at this 62th residue. So what is R? Well, we should remember all of our amino acids by heart. This is one of the abbreviations that is arginine, right? R stands for arg or arginine. And what do we know is special about arginine? Arginine is one of the positively charged amino acids, right? And so that's something that stands out to us, right? We have R changing, and that means a positive charge is changing, right? Delta in R means delta in the positive charge. So we're looking at something happening because a positive charge is changing. And so what do we know about positive charges, right? What do they like to interact with? They like to interact with negative charges. So if a positive charge changes, then it's not going to be able to interact with that negative charge as well. So which answer choice has to do with a negative charge? Well, not really. It might be crucial to protein folding, but that's not really what's being talked about, right? It's saying one amino acid is changing, and because of that, we're losing this inhibition. R62 interacts with phosphate in the anion binding pocket. Well, phosphate does have a negative charge, right? PO4 2-, very, very commonly used negative ion. C, R62. So I'm going to stick with B for now, but let's check C. Facilitates glutamate interacting with phosphate in the anion pocket. Well, glutamate is not mentioned anywhere in this sort of passage, right? Here it's talking about phosphate-induced inhibition, so we're going to eliminate C for that reason. D, R62 creates the only anion binding pocket in the protein. Well, we don't know anything about it being one anion binding pocket. There could be plenty of anion binding pockets throughout the organelle. So just because, remember, we're not trying to bring in external information on the MCAT. We're dealing with what we know on the paper. And so we can eliminate D2. And so B is our best answer, and we're 5 for 5. We did it. That one's a tough one because it's dealing with sort of, can we identify the amino acid being changed? Can we identify the effect of the amino acid being changed? As well as, can we make sure we're honed in on that, right? We're not changing our answer because there's other answers that might sound great. We're focused on what's being changed, what are the effects, and we get B as our answer. All right, great work, team. We did it. We crushed a biochem passage. Hopefully that was helpful, seeing exactly what's going on in my head as we tackle this section. If you like this video, please give us a like down below and subscribe to our channel, and comment what you want to see next down below. Also, check out our 528 series by Vikram. Perfect score on the MCAT and absolutely super helpful to get your dream score. We also have our MCAT question of the day, which we can send right to you. So check out that link down in our description. Happy studying, and hopefully that helped.